Dear colleagues, welcome to our contribution to the World Cancer Research Day 2021. Let's start with some breaking news. The cancer killer has been found. It has a name. Its name is P53. Newsweek made it to the title page. The molecule made it to the Molecule of the Year of the Science magazine. That was in 1993, 28 years ago. What happened since? What we knew already, or guessed already at that time, but now it is even more clear. Cancer causes, it's all about biology. Biology, it's all about regulation. Living systems grow. Cells grow. Cell growth has to be regulated, has to be tightly controlled. If cell growth is dysregulated, a tumor will be growing. What are molecules that are involved in such a mechanism, that are involved in a dysregulation of the normal cell growth? There are many molecules that are called proto-oncogenes. A single mutation in one of their genes may cause uncontrolled growth of a cell and a tumor. Some examples you can see here. All of these that I have selected are transcription factors. As you can see, they're all binding to DNA. There is the Jun and Phos family, comprising the AP1 complex, the ETS, the MYC and MYB factors. Of course, there are others that are not transcription factors, but these are very important ones since they are all involved in processes that are connected with cell growth, proliferation, differentiation, and so on. But there are others, we call them tumor suppressors. Amongst them is our good friend, P53. If these tumor suppressors that are deeply involved in controlling tumor or uncontrolled cell growth, that are involved in apoptosis, when they mutate and lose control, the consequences will be fatal. And as you can see, at least this one, P53, also is a DNA binding transcription factor. How is cell growth and our cell division processes regulated? It's a very complex process, especially in the cells of the higher organisms like the human body. One of the different circuits that are involved in that is shown in this diagram, which are all the cyclin CDK kinases that control quite a number of processes. And in the center of all these mechanisms, you see the E2F transcription factor. But that's not all. Also, the network where our P53 is in the center is an important part of all the network controlling cell division and growth. Some years ago, we put all these things together and added other processes that are important for cell growth. And then you end up with a network that may look like this. We put it together, more specifically, a very good friend and colleague of mine, Manabu Suji, from Chiba University in Japan, has put this together in a tool called Cell Illustrator for simulation purposes. And it worked quite nicely. He could even model then effects of individual mutations. So we have learned a lot about how cell cycle is regulated. And we have more knowledge about all kinds of pathways that regulate these gene regulatory processes and so on. But how can we make use of all this existing knowledge to deal with cancer as a disease? First of all, we have to prepare the knowledge that we have in a way that we can make proper use of it. From the experimental side, we have to record molecular profiles. We have to find out what are the transcriptomes, how the proteomes look like of a particular patient. The epigenomics, the metabolomics, you name them. Once we have these molecular profiles recorded, we have to explore them systematically with state-of-the-art computational methods. What is not state-of-the-art is to go with a pencil through paper-printed lists of molecules and see what might be interesting because of your personal prior knowledge. We need state-of-the-art computational methods, bioinformatics methods, to get the best out of the high throughput data on the patient's disease. Once we have these molecular profiles interpreted and got some insights, we have to connect these insights with clinical data. Today, we are going to show you a few of the tools and approaches we at geneXplain have chosen to deal with the mentioned problems. Obviously, geneXplain is a bioinformatics company. We don't do any experiments or clinical investigations by ourselves. For that, we work with excellent partners around the world. geneXplain is maintaining and distributing databases on gene regulation, especially with our database Transfact. You will hear more about the recent developments of that important resource. Databases about human and model organism proteins, their disease connections and pathways, as in our databases Human PSD and TransPath. And about enzymes in general across all organisms with the Brenda database from Braunschweig University. Of course, to make something useful out of the data and the knowledge we got, we have to apply state-of-the-art bioinformatics software. Our geneXplain platform is a huge collection of tools and programs that can be applied for all kinds of bioinformatic approaches. More specifically, the tool Genome Enhancer, and you will hear more about that in the next talk by Daria. And this is a very important tool to interpret omics data with regard to a certain tumor phenotype. Recently published and issued is the Match Suite as part of our Transfact 2.0 database. And this is something I will tell you more about in the last presentation of today's webinar. And last but not least, we have a couple of very advanced and highly powerful cheminformatics tools, PASS, Pharma Expert, and GUSAR. They will play an important role also when we deal with the Genome Enhancer.